Good evening, everybody. I'd like to invite you, I'd like to introduce Professor Ульріх Т. Schwarz from Technical University Chemnitz. I suppose that we will continue on our collaboration, and this guest lecture is the first step for this collaboration. Our students, Bogomol Danila is bachelor students of Theodor of Ульріх and also Vita Viktorovna, and everybody shows the topic of lecture, and I suppose it will be very interesting for us as for specialists in high technologies, as well as for specialists in science of living science. Okay, thank you for this short introduction. Please, Ульріх.
Yeah, Professor Monastersky, also thank you very much for inviting me to this seminar. And I mean, I expressed that I hope that we can repeat this soon, being in presence in Kiev. Right now it is difficult, as we all know. Yes, and it's a starting point. We are already discussing about collaboration, and we have Danilo here in my laboratory, which is wonderful. Yeah, let me just start into my talk. For the topic today, I selected one topic which is rather interdisciplinary between physics, technology, and also biomedicine. We have a startup here. This is something which we want to become a human medical implant in some time soon. We also work together with people from biomedicine, with medical doctors. So I have the names of the founders of the startup here, which is Tobias Moser, as a professor of medicine in Göttingen, and his former PhD student, Daniel Keppeler. My former PhD student, Christian Gosler, who did the microsystems technology development. My name is Ульріх Schwarz. Yeah, and it's about optical probes, which are made so that we can stimulate neurons in the inner ear. So you can also say they are made so that you can hear the light. And why is it important to hear light? I will come to this in a minute. But first, a very important disclaimer. So I'm in physics. I'm a physicist. My training is in optics, optoelectronics, and laser physics. So most of my research is on group 3 nitrides, which is wideband semiconductor. We helped to develop laser diodes and light-emitting diodes. We do a lot of spectroscopy, like on the top left, you see a typical part of an experiment, lots of optics. In the top center, you see a high-resolution image and spectroscopy by confocal microscopy. So these are the techniques which are mostly used in our group. And so we discuss carrier transport and recombination in in-gun quantum wells, and do semiconductor physics on the material and device level. We also have some projects on sensor science. About those I will not talk. Today I will do a lot of teaching in the direction of sensor science. And then we have this particular project on the optical cochlear implant, which is bridging our knowledge on the light emitters into the medical region, working here with people in Göttingen. And that's what I'm talking about today. So the outline, there will be a motivation And then I will talk about biomedical aspects of and optogenetics. Yeah, so please be patient. We will come back to semiconductor devices and their processing, but only when I arrive at the micro LED arrays and then waveguide arrays. Yeah, but we need the background first. Why are we doing that? What is it for? And it's about hearing. So maybe you know the conventional electrical cochlear implant, but let me just give you a few basics. So the acoustic hearing of a healthy ear is by mechanical stimulation of the inner ear. There are membranes here with hair cells. They can be stimulated mechanically, and then they trigger neurons and they send signals to the brain. And now in this cochlea in here, depending on the frequency of the sound, the hair cells and neurons in a certain place will be excited, and that corresponds to the pitch you are hearing. Good. Now there is something called an electric cochlear implant for people who lost the hair cells or there's any other defect which disables hearing in the conventional way. And what one is doing, that there's a hearing aid outside the brain on the ear. There's a coil, and behind the coil there's implanted in the bone of the head another coil as a receiver. These coils transmit the information and the energy, and then this receiver is an electric equipment. From there, you have a silicone cable going into the inner ear, and then there are electrodes here. Here you see it in a cross section. This is the silicon tube with the wires and the electrodes, and now electronic pulses are generated, and they stimulate the neurons by the electric potential, and the neurons then signal up to the brain. So it's possible to hear with such a device. However, this is limited to only about 10 frequency channels. Yeah, and that is because everything in here is conducting. You have a liquid which is filled with many ions in the body. And so when you stimulate an area here, it will stimulate a relatively large volume of your neurons. So the whole frequency range a healthy person can hear is subdivided into like 10 different channels, which is good enough to understand speech when you're trained, but it's very poor to distinguish between different speakers, to distinguish speech when there is background noise, and so on. So here you see this again. So these are these typical silicone tubes, which are then in the inner ear. This is an electrode, and these are the wires going from electrode to electrode. And basically what I showed here, I do have an audio demonstration, but it does not work across Zoom. But I guess you can imagine how sound could be when you just have 10 frequency channels. It's like a robotic voice, but very, very poor quality and difficult to understand, and no possibility to distinguish between speakers. However, this is still the most used or successful neuro implant. But there are more than 700 1,000 cochlear implants worldwide. It's a gigantic market. And in many cases, this is already implanted in the first year. So if you detect that a baby is deaf at birth, after birth, this is being measured. And then these cochlear implants are usually implanted in the first year. And they can be very successful, or they are very successful, and also big markets. So for the babies, usually you don't have a hearing aid at the ear, but you have it somewhere clipped to the cloth, or yeah. Good. Yeah, and now what can we do to improve that? And this is where this Professor Tobias Moser from Göttingen addressed us, and he had the idea to stimulate with light. So instead of electrodes, to implant a string of light sources and then excite the neurons with light. And the advantage is that light can be confined to a much smaller volume. I don't say focused, you don't have optics in there, but it's not scattering into such a large volume. And then you can excite not only 10, but more than 100 channels in the ear, frequency channels, which is kind of the high fidelity version of a cochlear implant, highly improved. You can understand music, you can distinguish speakers, you can hear pitch. It's just a factor of 10 at least difference, which is much, much better. Still, first you need to make the neurons to react to light. We come to that later. You of course need to make such a light source which goes into the ear. And this is of course the major topic of today's talk. Here again, I would have two simulations, but they don't work via Zoom. I think I have something here where at least you can visually see how it should be. Again, there's sound, I can hear it, you can't hear it, it's Beethoven. But the idea is again, in the cochlea to have a light chain, which is then communicating sound and the different sound channels. Good. How do you make the light sensitive, this neurons sensitive to light? First, Yanadi, can you hear me? Everything is okay, language, or your microphone is muted, but I assume it's okay.
Yes, it's always okay.
Good, great, good. Also, if there are important questions in between, just ask, but I guess we will have a discussion session at the end. So how to make light, neurons sensitive to light. And this is a very important field in biomedics, medical. So it's called optogenetics, and some ion channels, which are original from algae, are implemented into the outer shell of the neurons. This is like shown here on the right side. So if this is kind of the cell membrane of a neuron cell, then these are special big molecules called ion channels, and they react to light, open and close in response to light, and then trigger first an ion transport across the membrane, and this changes the potential across the membrane, and which makes the cell to trigger and give the electrical signal which a cell is neuron cell is made for. So again, this is like shown here. If you have a neuron with these channel rhodopsins, that's how they are called here. And this is the prototype, the channel rhodopsin 2, mostly used, reacting to blue light. If this is implemented here, then you add blue light, and then the cell will fire. So this is again shown here. This is a patch clamp experiment. So this shows the potential, and after a light pulse, the cell is just firing. There are many details on this. It's a very successful method used in a large number of laboratories worldwide, in particular in neuroscience. And so there are people developing different channel rhodopsins, slow ones, fast ones, reacting to different wavelengths, and also methods to implement them. And like typically in genetics, you can address certain cell types. So that's like also shown here. If these are two different cell types, like in the brain cortex, then you can specifically attach these ion channels to one cell type and not the other. So be selective. And yeah, good. This works. That's again Tobias Moos and Göttingen, because we don't do biomedical here, medics here. This is a cross-section of the cochlea. You see the channels. Red is the bone, and green are the neurons with the channel rhodopsin expressed. Yeah. And here in the center, you see an experiment where in the brainstem, the signal is picked up electrically, and then here red marks an acoustic click, and you see a response. Here blue marks an optical excitation, and you see a response. So the neurons do react to light. Okay, this is another experiment now in an animal, in a mouse. So you take a virus, an adeno-associated virus, to make the mouse neurons express the channel rhodopsin. In the center, you see that this works. So these are neurons again. The blue stain shows is a marker. Also here, you have markers which indicate that indeed the channel rhodopsin is implemented in the membrane of the cell. On the right side, you see how fast they can be, and this is important for hearing. Hearing is a pretty fast channel. I mean, to stimulate a 20 or a 2 kilohertz tone, you don't need to stimulate with 2 kilohertz. You need to stimulate the ear in the place where the 2 kilohertz tone is. But still, it has to be fast to allow you to detect directions. And again, hearing channel is pretty fast. And so what you see here, this is triggered in regular space, like 20 hertz triggering, and black response picked up with a probe in the brainstem, and this is very regular. So this shows it can clearly follow 20 hertz. It can also follow 100 hertz. Even 300 hertz is regular, and in the region of kilohertz or so, it starts to become more random. So this shows that you can do a true stimulation up to the kilohertz range if you have a fast switch, which is important for hearing. This is another and almost one of the last now on this biomedical side, important slide showing that we can, with the optical method, we can reach a resolution similar to acoustic stimulation. So what is done here is, again, a mouse is first implemented the channel rhodopsin in the neurons of the ear. So it reacts to light. Then by fibers, light is injected into a certain part of the cochlea. So here you see the cochlea. These are the windings of the cochlea marked. And then in the brain, where it is tonotopic, you pick up the signal of the neurons. And so these here now from top to bottom are the electrodes in the brain. And this is here sound intensity. And now with a single 4 kilohertz tone, only a small area in this brain region is excited, showing that you have the high frequency resolution, and with increasing sound pressure, the intensity, the response strength becomes stronger. So this is acoustic hearing. On the very right side, you have this with an electric implant, cochlear implant. And what you see is if you excite, almost the whole cochlea is excited. I mean, it's a small mouse cochlea, but still, so the spread is large, and also the dynamic range is very small. In the center, you see it with light. So again, you have the light intensity, and you excite here like in one space, and then also the cortex, only a narrow region is reacting, and also the dynamic range is pretty strong. So with light, we should be able to do something which is close to natural sound. Yeah, there are some publications to that, also more recent ones, if you're interested. And so that shows that, I mean, we can do this precise optical stimulation, so it should be possible to make a cochlear implant which is much better than the one which is used now with electrical stimulations. Good. Now comes our task. So the biomedical side kind of works. Also, there has still to be many things to be done to bring it to a human implant, but it works. On the optoelectronic science, we need to make a chain of light sources which fits into the ear of a mouse and later into the inner ear of a human. So we want to have like 100 light sources. They should be individually switchable, highly efficient, and they have to be small enough. So in the center, you see one of our first examples. In the background is the fingertip, and in the front here, this small thing, a few millimeter long, 5 millimeter long. Here you see it magnified in the bottom. This is a flexible string of micro LEDs, and a single micro LED here is illuminated. So that's the size we are talking about. Yeah. Usually I'm joking, it's like this Christmas string, which you find now everywhere for the Christmas tree illumination, but just for the tiniest of tiny toy trains. Yeah, good. It has to be flexible, so it goes into the ear. So that's shown here. It bending radius of 0.5 millimeter to 1 millimeter. This would be for a rat cochlea. And this, again, that's the first example where we had 25 LEDs and we could only switch between every second one. So not yet single addressable. Good. Another very, very difficult condition is that it has to be biocompatible and it has to be stable in a body for about 80 years. And we come to that also at the very end. So if you talk about human implants and you implant it to a 1-year-old, you cannot and you don't want to replace this. Good. So the technology behind that is of course, these high power, high efficient blue LEDs, which are used in lighting for general lighting, yeah, white lights. I hope you are aware that white LEDs don't exist. So it's always a blue LED plus a phosphor, which makes it white. So here you see the blue LEDs, and then this, in that case, the phosphor is in a plastic shell, so that makes it white. So we worked on this technology, and now we have to implement it to go into such a small probe. Yeah, and small means small. I mean, this here is a standard small LED, so kind of a tiny one, which is still commercially available, 200 by 200 by 200 micrometers cube. That would be the central contact, and that is a hair. And this is another neuroprobe, about the same scale. And you see, this is much too big. That's not what we can need. It has to be flexible. So the only way is that we just take only the semiconductor layer and also make it smaller to be able to implement it into a probe like this here. Here in the background, you see neurons. Yeah, so that's the size we are talking about. And what we did is that, yeah, also what we want to do is we want to make this a commercial product. So from the very beginning, we were looking to make a process which can be scaled into production. Yeah, it's not helpful to make a single device. We need to develop a technology which can be upscaled. So here we have a wafer level process. Yeah, it's a 4-inch wafer. And you see all these golden lines here. These are individual cochlear implants, which can be later removed from the wafer, but the whole process is on wafer scale. This was still done when I was working in Freiburg. And so the clean room where we did this was the IMTech, together with Patrick Ruther, Institute of Microtechnology in Freiburg. The project name was Lichthören, which means hearing the light. And now I'm show you some details here on this technology. So it's all microsystems technology, wafer level again. So we start typically with like lithography. On the right side, you see a mask for the maser of the LEDs. So that is the 4-inch diameter here. And all the small spots here are LEDs. Here again, so the blue are LEDs on the probes. And of course, we need several mask sets, several level, the typical thing what you do in microsystems technology to build a device. And the process which we have here starts on one hand with epitaxy of the laser, of the LED wafer. So you need an efficient blue LED. We have it here on sapphire. So it's a gallium nitride on sapphire LED epitaxy wafer, 4-inch again. Blue is the wavelength. And then we do a gallium nitride on sapphire LED process to make the individual LEDs. On the other hand, we make a polymer and metal on silicon or on sapphire carrier wafer for the contacts for the other part. Then we combine them. We do a laser LED transfer and a laser liftoff to transfer only the active 5 micrometer part, gallium nitride part with the LED onto the polymer. Then we do the second contact layer, the top, and up to here it's all on wafer level. So this is still all on a 4-inch wafer, and then the wafer level process stops and the individual probes are removed, and we still need to do the electrical interface, contacts, and so on. Good. Now some details on this process. So on the left side, you see the LED wafer. It's a gallium nitride on sapphire. So it's a thin film LED process. You have a sapphire wafer. You have a N-gun, P-gun, and active layer, of course, which is not shown here. So in the center, you have the quantum wells, electron barriers, and so on. So this is the epi stack of an LED. And then it is etched down. You have a mesa and the chip. It's etched down by reactive ion beam etching, hard mask technology, to make to make the individual LED chips. They have a typical side length of 50 by 50 micrometer. The thickness is, as I said, about 5 micrometers. So this is not to scale. They're actually pretty thin. Good. Next, you need metal contact. So you have a P metal, there's a diffusion barrier, and an indium for bonding. Yeah, good. And last, you have a sidewall passivation here to insulate the sides and also reduce non-radiative recombination at the sidewalls. So this is one wafer, the wafer with the LEDs. And then you have, again, a silicon or a sapphire. You have the other wafer, which has a flexible substrate. You have a metal layer for contacts, and then also bond, thick gold for bonding. So indium and gold, this makes then the connection. You need the transfer because the epi has to be done on sapphire, but sapphire is not flexible. So that's why you need to transfer this to that side. Good. You need the diffusion layer because when you do the bonding, you don't want the indium to diffuse into the P metal. So there are many, many details. Now for the transfer, you take the 2 wafers and then you flip this on that one. And then you do here a bonding by pressure and temperature. And many steps omitted now. After bonding, you add an underfill here to stabilize it. So between the 2 wafers, you add an epoxy. And then the sapphire is removed. This is done by laser liftoff, which means you shoot with a ultraviolet laser, excimer laser through the sapphire and evaporates this interface between the sapphire and the gallium nitride. And then the sapphire can be removed, leaving this plane open. Yeah, and then again, another metal layer is added to contact the N side, and another polymer is added, and you open the contacts. Now you have here a full LED, N side up, P side down, metal layers for the contact, for the bonding, and here are the metal wires and the 2 contact sides. So this ends the process. Again, here are the steps. So as I said, you start with the epi, you do the P metal, you do the etching, you do the sidewall passivation, you add the bond metal here. On the carrier, you start again on silicon or sapphire, the polymer metal, the bond metal, then you do the transfer. So you transfer one layer onto the other. This is bonded by temperature and pressure. Then you do this underfill here. Now it's still a sandwich. So sapphire on top, sapphire or silicon on the bottom, and in between you have your probe. And then with the laser, this interface here is evaporated, and you can remove the sapphire, and now you have only this layer. And later you will pull it off from the silicon, and then only this very thin layer here will remain. So after this step here, you add the N contact, you add the another flexible polymer here, and the contacts here. Now you have a thin, very thin and flexible probe if you remove it from the silicon. Yeah, that's how it looks like. So these are probes now with 144 micro LEDs. You can address them individually. Each has 50 by 50 micrometer size. Each of these dots here in the center, and the pitch is 100 micrometer, and it fits into the cochlea. And this shows that one can just be illuminated by contacting the backside with the needles, and you can go from one to the next here. And this is the end. Here you see also the needle for the contact. So you have all individual LEDs being addressable. Good. Yes, and this is a whole chain of them wound around a stick with 1 millimeter. It's a glass rod, 1 millimeter diameter, so it's very flexible. This is when they are illuminated. They're shining inwards. That's why it's kind of dark here and only bright on the outside. But here, if you look from the inside, it's bright, as it should do. And it is able to fit into the cochlea. So this is a micro XRD tomography, so X-ray tomography of a cochlea. And this purple ribbon here is the implant, and the spots are all the individual LEDs. And this here marks, the color marks the place of the neurons, of the nerve cells. Yeah, and the membrane here in the center. So it nicely follows the neurons. And now you can imagine that by switching on and off these LEDs, you can stimulate the neurons along this membrane. Good. Yes. Now, so that's kind of how in the end, what we want to have for a human. We want to have for human an implant. So again, that is the external hearing aid with a processor, with a battery, and then you have this magnet and coil. This is the part which is implanted inside the skull bone, and then there's something going into the inner ear. This is the front part, and that should be in the cochlea where you have the light emission. And of course, you also need the optogenetic parts, which brings this light switches into the neurons of the inner ear. You also need to make sure that this is safe in terms of medical means. Good. However, and this will be now the last part of my talk, we realize that it is not possible to make this micro LEDs stable for 80 years. I mean, they are in silicone, but it's never hermetically sealed. Water will go into this part, ions will go into this part, and there will be corrosion. So it's not possible to have this as a long-term implant inside a human body. It's very good for preclinical tests with mice, and it's being used for that, but it will never be a human implant to our knowledge. So now we slightly changed our strategy here, which is we are looking for something where there's no active light emitter in this part. So now we have the active light emitter as laser diodes inside this hermetically sealed package here, which is titanium and glass and ceramics. And then outside only polymer waveguides. So that's what we developed recently here in Chemnitz. And here you see like the tip of such a probe. Again, these are light emitters, but now as outcoupling structures at the end of waveguides. So these are waveguides, and I can switch here. So by coupling into the different waveguides, it's outcoupling at different places. So it's doing the same thing, but now we don't have an active electronic part here, but we just have scattering structures in this place. And there's only a polymer going into the inner part of the ear. And this is still a wafer scale process. So we're not changing that. Now it's a 6-inch wafer here. Here again, you see these probes. That's the design. So at the tip here, they have this outcoupling structures. That's a detail of an outcoupling structure. Then we have long waveguides, which have to be low loss, low scattering loss. They have to be flexible again. And then it's fanning out here so that we can couple into these facets here from individual laser diodes. And depending on where we couple in, we will couple out here at different spaces. This is scanning electron microscope image of one of these tips here. So you see this coupling structure. This is the waveguide. This is a close-up and cross-section of a waveguide. So it's an SU8 waveguide and then PMMA cladding. Yeah, it's moderately thin, so it's very flexible. And this is how it looks in reality. So just cut out here a string of these here. There are the outcoupling structures. And in this housing, you have a laser diode which couples into the waveguides, and so light is coming out at the end. This was also first prototype. This is an example where the light is now coupled into one of the waveguides, into the top waveguide. So light is coupled out here. You see there's some crosstalk, so there's a little light in the other waveguides too, but this is a logarithmic scale, so it's 2 orders of magnitude lowers. So most of the light propagates in one waveguide, and you can also switch between different waveguides here. I just have 2 pictures in that case, but you can just switch all the waveguides. Yeah, so that's the technology which we are following right now, which has the potential to become a human medical implant. Good. Okay, with this I come to my summary and hopefully to discussion. So I showed you in the beginning how we can switch or stimulate neurons and control nerve cells with light. And the blue LEDs and laser diodes are enabling technology for optogenetics. Most of the people still work with channel rhodopsin 2, which needs blue light. We're also now working with other rhodopsins, which work with red light for some reason, but we need the optical light source for that. I showed you the cochlear implant, how the standard cochlear implant works, and how we want to use an optical cochlear implant for high fidelity. We did this first with flexible micro LED arrays, developed a special thin film process for that. And maybe one thing I didn't say, so the really new thing here is that we have to develop many new process steps which are compatible with the polymers. Yeah, I mean, of course, thin film LED exists, but only with rigid substrates, which can do high temperature processes. But now you need to do these processes for low temperature, so that you're not damaging the polymer. Good. And in the end, I showed you kind of the next generation optical CI, where we're working right now with waveguides arrays. And the vision is to make something which can help humanity as a medical implant in some time soon. And with that, I thank you very much for your attention Thank you.
Thank you.
Thank you for your presentation.
Thank you very much for very amazing presentation, Professor Schwarz. And please, question, if if you have question, please don't hesitate.
Well, may I ask some
Okay,
question?
okay.
Yeah?
Yes, Vita,
Yes.
please.
Yes. Ульріх, I have some questions, minimum two. For the beginning, first of all, can you open slide number 11 and explain some pictures?
Mm-hmm. Yeah. Oh, okay.
11.
Yeah, this is 11?
This is 11.
No. Oh, no, not 11, up.
Or other direction?
No, down. Oh, this.
Okay.
No, up.
Previous.
Yeah.
This picture.
Okay,
yes.
good. Okay. Yeah.
Yes. Am I right when thinking that light stimulation is preferable comparing with current or electric or sound stimulation? Is it more clear or effective? Because I think that sound stimulation is natural for audio hearing.
Yes, so let me put it that way.
Why light? And the second part of
go on.
this question, why blue leads, LEDs and light LEDs?
Yes, okay. So for the first question, I mean, we don't want to be better than the human ear with sound. So if you have normal hearing, keep it. Yeah, we will not implant anything, of course. We talk about people who are deaf, who can't hear. Yeah, and this is affecting many people from birth on. I mean, many people are born without being able to hear. And also nowadays it is standard then when a baby is born that you there's a certain device, you put it into the ear and you push a button and then it will tell you if the baby can hear or not. Without the baby has to do nothing. Yeah, this would be another story. It's very interesting, but this really works. So you will know if a baby can hear or not from the first day usually. Yeah. And so if a baby is deaf, now that's what we're talking about. Or also adults. I mean, if an adult is losing hearing completely, I mean, if it's only partially lost, you use a standard hearing aid. Yeah, but if there's complete loss, so that a hearing aid will not work, a standard hearing aid, you can still use this one. Yeah. So sound is not working anymore. Yeah, and now we have the electric implant, and this is kind of has very low frequency resolution. And what we show here with these three pictures is that with light stimulation we can reach a frequency resolution which seems to be almost as good or similar to sound stimulation, yeah, but much better than the current the electrical stimulation which is currently used. Yeah.
Ульріх, please.
And about blue. Sorry.
Yes. Okay. About blue. So
the next part.
Yeah, the next part I have to go. I have an extra slide for that. So. As I said in the beginning, many people work on different of these molecules which do the switching. Yeah, and for example, here on the left side, you have a map that's from 2015, where you have all the different molecules which are being used. For example, the channel rhodopsin 2, which I talked here, which is sensitive to blue. So here's the peak activation. So blue is a channel rhodopsin, and there are 1, 2, 3, 4, 5 different variations. And this is the switching time, how fast they switch off, and which also tells you how fast they react. Yeah. And so, and there are others which are working red. There's fast ones, there are slow ones. This here shows how they kind of the wavelength response, wavelength and time. So why blue? So the channel rhodopsin 2 was the first really successful one, and it's being the one which is mostly used in the laboratories worldwide. So for preclinical studies, this is really the major thing. So many of them are blue. Yeah? However, if you go to the human implant, there's a danger that when you excite neurons with blue light for many, many years, that the neurons will degenerate. Yeah, like it's a phototoxicity. So this is why we now try to use a different one. We use this crimson here, or crimson variant, which is excited in the red wavelength region, and which is fast. Yeah, but again, this is like one is for the human and the other is for the preclinical study.
Okay, thank you. Thank
Welcome.
you.
Thank you. Rita, one more question?
Yes, can you show 26 number?
Yeah, here.
No, next. Oh, here.
This
what is the distance between images in flexible with waveguides device?
Yeah, so down here you see a scale. So the distance here along the probe here is, yeah, in this case it was, I think, 200 or 250 micrometer. Yeah, we want to have several per millimeter, certainly. Yeah, factor 10 better than the electrical one. And the distance or the pitch of the waveguide is about 50 micrometers. So between the waveguide, it's 50 micrometer. Yeah.
Okay. Thank you.
May I ask a question?
Yeah.
Yes, of course.
How to deal with the removal of heat of such a small scale from the LED so that it doesn't change its characteristics?
Yes. So, I mean, here inside, there is no loss and no heat generated. Yeah. Also, the power is very little, which we need to stimulate in the inner ear. However, the laser diodes, of course, they need power. Yeah. Still, we talk about small laser diodes, and I mean the excitation is pulsed, so that the overall power which has to be dissipated is is moderately small. Yeah, and also the power will be so the power is generated here in this housing, which will be here in this bone behind your ear. Of course, you cannot heat it a lot, but I mean, if you generate a few 10 milliwatt in this and then dissipate it through this big housing here, this is still tolerable. And you're not exciting the laser diodes CW, but this is with pulse generation to increase the efficiency, short pulses, and yeah.
Okay, thank you.
May I ask a question?
Yes, yes, of course, please.
Okay, I have two questions about safety. First of all, safety technical, yeah. So, do you have some algorithm that protects you from hacking from outside? For example, if someone puts a directional sound wave on your device, and it will activate all of your laser diodes, yeah, simultaneously. It will mean that people who has such device in your ear will hear something terrified. Yeah. Did you think about like, do you have some maximum output power and maximum active diodes at once, something like this? And second question is about safety biological. Yeah. For example, if you put some molecules that induces ion conduction in your ear, yeah, in a baby. So it means that this baby will rely whole life on some foreign molecules that will produce this conduction, yeah. And in theory, it can influence its DNA in future because it's like will change it in some way. I know it's like way too far.
Yes. Okay. And, no, fine.
Yeah.
So let's start with the first one. So a few things. So first, I think in the conventional cochlear implant, there is. Yeah, no, it's not a secure link. I mean, in principle, you could kind of send in some wrong data. Yeah, you could. I mean, if you could envision that somebody is really putting in some wrong data. I mean, there is of course some security in terms of at least how do you say? You're checking if the data is corrupt, of course. Yeah. But it's not a high security link, yeah. So you could also envision that a standard electrical implant, you by some way you can electrocute the person, yeah. However, this is impossible or highly unlikely because the power which is transmitted into this device is really limiting what is available. I mean, it would be difficult or impossible to get enough power out of all this laser diode to do real harm inside, just by the power which is available through the through this coil. And also, I mean, of course, inside here there will be an ASIC which is doing the control of the lasers, and I mean we haven't developed this yet, but this will allow only to switch a single laser one after the other, and limits kind of the time which is as it is on. So there are more easy ways to harm a person than by hacking this device. What is possible is that you can communicate with a person without anybody else understanding. Yeah, I mean, this is also nowadays possible. So a person which has a cochlear implant, if you have here a connection, and this is not only doable, this is existing. I mean, they have Bluetooth available. So a person with a cochlear implant, you can transmit some text to her, some speech to this person, which is not being heard by anybody else. Yeah, I'm not sure if this is being used by somebody, but it's possible technically, easily. Okay, to your second question, this is really the big one. So, I mean, of course, I'm holding my breath when I speak to this medical doctors about first doing experiments in mice. Yeah, I'm happy that's not what I have to do, but I think it's always necessary. If you want to develop a medical implant, you have to first work with preclinical studies, which means with animals. You try to minimize it. And you have to guarantee safety for humans. There are all the standards there. And anything genetic is super critical. And there's a big question if this will be accepted. So will anyone accept this technology? I mean, a much higher quality in hearing for deaf people. Will this be accepted on the risk of allowing optogenetics? And this is a big question right now, also in other case instances. However, there are first medical curings out or treatments out which are based on genetics. Yeah, on genetics. Yeah. Recently, there's one curing, I forgot what it was, but it's super expensive, but it's curing a certain illness. And actually, with the eye, there are studies using optogenetics in the eye. And that is because there's nothing else possible in the eye. For the blind, there's no implant which will help. And so they try to get something done there. For the ear, we have the competition with the existing implants, so we have to be much better. And then the question will be, will it be accepted? Regarding a modification of the human genetic kind of line, this is, as far as I understand, not an issue. And that is because of the way how it is implemented. Again, it will be through a virus, which is actually almost the same, or the same one which is now used for a COVID vaccination. Yeah. So this is what you all already are doing. I mean, also like a flu vaccination. These are similar technologies. So we are not talking about modifying the DNA. Yeah, we are talking about putting in RNA, which then makes the cell build the switch. Yeah, and because it's a neuron, so it's a nerve cell. Nerve cells are permanent, so it should be sufficient to do this once in a lifetime. And there are data which shows that it's likely to work. Yeah, but we are not talking about modifying human DNA. Absolutely not. Did this answer your question?
Yes, of course. I wasn't talking about directly modifying the DNA, but the modifying of the DNA by making the human rely on such implants for a long time. That's why.
Yeah, okay. Yeah. But that's another topic.
Yes.
May I ask one more question about technical part of this device? As I got clear, if I got it clear, you have like lithium-ion accumulators there. And what is the capacity, and how long can a person use this device on one charge, for example?
So we are targeting at least 1 day at the time being. So what you have to do is, like here, the lower part is the battery. And I mean, changing the battery more often than once per day is not convenient. Of course, we want to have it longer lasting. I mean, the first devices will not be the most efficient, but also the electrical cochlear implant, it was invented, or not invented, it was first implanted in 1979. And it took also some time to develop. Or think about a heart pacemaker. I mean, the first heart pacemaker, they were huge and you had to carry a battery. And I mean, we are not as bad, but we are lucky if you can start with 1 battery per day for the first 20 patients. And then, of course, one has to improve the technology. In principle, it's feasible, but there are also many unknowns. We don't know how much intensity we need. also like when we look at this picture here, I mean, a lot will depend on how efficiently we can couple the light from this emitters to the neurons, yeah, how the geometry can be done. Right now, we are happy if we just get it somehow into the ear. And then in next generations, this can be optimized. Yeah. And then maybe, but in principle, it can be compatible to what is being done now. But it's a super important question, yes.
May I say one more thing?
Yeah.
About the orientation, I heard that there are, like in area on this picture. Currently, you haven't the possibility to make this, but I heard that there are some type of clothes which can be make more hard or more soft by electrical currents. So in theory, you can like rotate it with making it more soft or more hard, and it in theory can possibly help you.
Yes.
To...
Yes. If you're a specialist in that.
No, I'm not.
Okay, because something I'm also looking for is like when we couple into the waveguides here, when we couple into the waveguides, this needs to be very stable. We talk about 80 years, and there's no way to make an alignment. And so I wonder if something like a polymer or another material exists, where we can have like a tiny attenuator, like only for like 2-3 micrometers or maybe 10 micrometers, where we can shift our laser diodes. Of course, it has to be stable, and it's not allowed to use a lot of current. But that would be nice to have a very tiny, like I think of a polymer which you can expand by adding a voltage and without having too much current. But yeah.
Well, I know for sure such thing exists, so you can search for it.
Yeah.
Okay, just another question. I want to expand first question of Sir Demidov. Wouldn't external electromagnetic fields cause enough fluctuation in the device to activate LEDs of such a low power?
I don't think so, because I mean, of course, it's modulated. So you're working on a certain frequency, which is, I think, in the megahertz regime. And so like a standard low frequency magnetic field has no effect. I'm pretty sure that when you're kind of next to a Tesla coil or so, maybe you don't do that. It's better not, or other things. So I mean, if they're strong and high frequency fields, I'm pretty sure it could have, of course, an influence. And the first influence would be that you have like a lot of errors in the signal, and then I mean, of course, the active device inside will just tell you, no, okay, this is not a valid code, ignore it.
Okay.
Yeah.
Okay, one more questions, please.
One brief question. What about influence of environment on these implants?
Yeah, again, this is so critical. So, I mean, this top part here, where the electronic and the laser diodes is, this is hermetically sealed. It's ceramics, it's titanium, it's welded. So that should be safe. It's like a heart pacemaker or existing devices. So we know that this works. I mean, this is like in a standard cochlear implant. And down here, it's only polymers in silicone. So there will be humidity will go in there. There will be a balance after some time in terms of diffusion. We still need to validate that this has not a too bad influence on the light conducting properties, on the waveguide properties, but there's no active device which can be damaged, no current, no voltage, so it's only the polymer waveguides. And also, I mean, in the other direction, of course, we have to make sure that it's biocompatible, so that there's nothing which can go out and be bad for the body. So the polymers have to be selected. Silicone is possible, medical grade, and PMMA is possible. For the SU8, it's being used in many preclinical studies, but as far as we know, it has not yet been certified for human use, medical applications. So this is still something to be done. Yeah, so it has to be sure. This is also a major topic, that both the body is not doing damage to the implant and that the implant is not having a negative effect on the body. Yeah.
Okay, thank you. One more questions, please. So I have one questions, technical and was more global. You told us that electrical implant have just 10 frequency channel.
Yes.
And the much most advantage of optical stimulation is the possibility to provide much more channel.
Yes.
Please explain more in detail what you mean. You mean that this 64 independent channel in human, which we saw on this slide, correspond to some particular frequency, or this is another mechanism?
No, yeah, no, you're right. So this is the cochlea in the inner ear, and the pitch, so that the height of the frequency is coded along this cochlea. So the lowest frequency are the top, and the highest frequencies are here. Yeah? And like if you do an acoustic stimulation here, kind of these membranes are vibrating. So like there are two windows here, and the sound is being coupled in, and the sound propagates up here and then down the other channel here. And by the change of the geometry, there will be a certain region where it is resonant. So if you have like a, let's say, 4 kilohertz tone, it will excite the membrane and this location here. Yeah. And then this here will vibrate in this location, excite the hair cell in that location and the neurons in that location. And this goes to the brain, and in the brain, it ends up at a certain space again. And after that, there are all the other layers which decode and speech and language and all this. But up to the brain, there is what I call tonotopic. So there is a space is mapped, or frequency is mapped to a space, to a location. Yeah. And which means if you excite neurons, let's say neurons on one space or the other, it will end up in the brain like a different pitch. And again, the problem is with the electric, you excite a whole range. And with the optical here, we can excite a much smaller region along this membrane. So instead of exciting a whole octave, we excite a small. So Tobias Moser says, like, the electrical cochlear implant is like playing piano with your elbows. Yeah. And we want at least to use our fist. I mean, maybe not a finger, but we can play it with a fist. Yeah. That's what we want to achieve.
And physically, it is realized by just by different intensity of LED. Yes.
Yes, by a different intensity of the light. So if you switch it on, intensity or pulse length, yeah. Particularly for the laser, we have one intensity, but we vary the pulse length, and this then triggers the neurons. Yes.
Pulse length. Okay, okay. And 64 is the most channel which is possible at the moment, or probably much more.
So in the electric, it's
No,
8
no,
to
optical,
10,
optical.
and in the optic, we hope. I mean, we don't know what is possible, but technically, we try 64 and 128. These are the numbers. I mean, we will have arrays of lasers with hopefully 32 lasers. And then if you have 2 of these arrays, then we are at 64 channels. 128 channels is technologically another challenge. We need 4 arrays. It's, I mean, scaling this up, it is possible, but difficult. So again, we hope to start with something which is much better than the electric. So it has to be better, because otherwise we cannot sell it. But I mean, once this really starts to operate, there will be enough money to scale it up. We will need different laser diodes, we will need different waveguides, but that will be possible. Yeah.
Okay, thank you. And other question, can you present some other
question? Are they able to listen to the music?
What do you mean?
Are they able to listen to the music with such Cochlea implants?
I hope so. Yeah. I mean, at least much better. So again, it's a pity that I cannot play the sound here, but it is kind of music. Of course, it's not high quality, but it's you get some of the beauty of music. Yes.
Okay.
Yeah.
Thank you. And other questions? More.
May I ask one question?
No, because I ask
first.
Now I am asking. One more question is the following. Do you present some other example of optogenetics? I mean, where is optogenetics?
Yes. So again, it's the tool of choice in neuroscience. Yeah. So it was, I think in 2014 or 2010, it was voted by Nature as the method of the year. So in neuroscience, it's also an old number, but like 1/3 of the laboratories worldwide work with optogenetics. So if you, this is, yeah. So it's definitely a very important scientific tool. For the human, other things could be epilepsy, Parkinson, heart pacemaker, and also for the larynx stimulation. So these are things which are right, and the eye. Yeah, so this is where this is being discussed, or where already other people work. Yeah.
Okay, thank you, thank you. Petro, you can ask if
yes, thank you. May I show the slide where it is maybe 20-someone, where you show the real form of waveguides? Next one.
Yeah.
And next one. Yeah, this one. So you know, previously you showed the slide number 7, and I thought that the shape of this thing should be more not like a plane, but more like a worm, you know, like some cylinder with segments.
Yes.
And
okay.
each segment should eliminate 360 degrees. So in this case, you have like, you don't need to position precisely
yeah, but you will also lose a lot of power. So what we want to do is that the light is going to the inside. Yeah, because there's where the neurons are. I mean, you fit it into the ear, and if it goes, then follows the spiral, then they shine in the right direction. Yeah, and this I think can be done mechanically. And regarding this one, this was just cut by hand. Yeah, this is very crude. I mean, this was, so we did the processing, and then for the demonstration, I mean, you see that here. This is just cut by hand. I mean, now we have another process to etch the shape of this one. I mean, when you go back here, no, I don't have a picture here, but now we can, like, we etch the samples and then we remove them. Yeah. So this was just the very first demonstration, but it's thin, and then it will be encapsulated into silicone, and then it's round again. So so we will have something which is round cylindrically, but we want to have it bent into a certain direction so that it follows the cochlea, and the light is going to the inside only.
I got it now. Yeah, thank you.
Yeah, welcome.
Thank you. One more questions, please. I have one question about the reliability of this micro LED.
Yeah.
How long it lives? You talk us about 80 years.
Yes.
But is it possible for LED to live so long?
So again, the LED, we give the LED up for the human implant, not because the LED will be stable. Yeah, we have it low power, and it's not operating at CW, but it's just pulsed. Yeah, so the gallium nitride LED, that will be stable for 80 years, but not the LED array. Yeah, so this is why we go to the waveguides. So this is only polymer, and I mean, we need to do more testing. This is very crucial, but that should be stable for a long time. Like, I mean, also silicone in the body is stable over many tens of years. Yeah, and also the standard implant is stable. I mean, the first implants were implanted in the late 70s. So they are now already 40 years in place. And also others are many years in place, so they are stable. And the laser diodes, which are then inside this housing, again, they are not operated CW, but only pulsed. And also the electronics, I mean, yes, it's critical, but like in the other implants, or like in a heart pacemaker, it seems to be doable. Yeah. But it's super critical question. Yeah.
Okay, thank you very much. Dear colleagues, please questions. Students, don't hesitate if you want to know more than you know. Please don't hesitate. Okay, we are working about 1 hour and a half.
Yeah.
And I suppose that it was very interesting topic, according, especially for Petro, the middle. This is our young teacher.
Yes.
A PhD, and as far as I understand, Petro also his interest concerning the biosensors. Yes, Petro?
Yeah, it's nanoplasmonics and biosensors.
So please just send me an email and contact me and give me some information. Yeah, why not? Yeah.
Okay.
would be nice. Yeah, to know.
I saw Vitaly Maslov. Vitaly Maslov is our colleague from Institute of Physiology. Do you have questions, Vitaly? No, not here. Okay. So thank you very much. Thank you very much. It was very interesting and very amazing talk.
Okay.